Hello everyone. Today we're going to talk about meiosis and mitosis. This is a screencast lecture that is designed to support your reading on this topic, and it's designed to provide sort of a personal take on why meiosis and mitosis are important and what are they and what's going on there. And so what I'm actually going to do with you today is take you back through a lecture that I gave in class recently, and I'm looking at my notes, and I think the best way to go back through it, I enjoyed the lecture, so I'm going to go back through here and I'm going to use my highlighter to do things like highlight meiosis and mitosis to let you know I'm right here with you, going back through these notes to talk a little bit about these ideas. First, I'd like to say that as you probably remember, you might remember if you've done the reading of cell division before this, that meiosis is making gametes, right? The gametes are things like in humans, these are like eggs and sperm, all right? And they are specifically made for making the egg, right? So they'll each contribute some DNA to this thing, not the egg, but this will make a thing called a zygote. And the zygote then divides and becomes an entire organism eventually, all descended from that single cell. Mitosis, on the other hand, is a little bit different because it is a method of cellular replication done by organisms like bacteria. So there are entire organisms that do it, and there are also cells in our body that do this. And mitosis is something that is used for what we call tissue growth. So if you have a tissue like the skin cells you have, for example, if you want to grow more skin cells, you use mitosis. If you need to grow more liver cells, you use mitosis. And those make replicates of the original cells, which is very different than making a gamete. And we'll get to that exactly how that works. So here's an image here of I'm going to use my highlighter to look at this little section right here. What I'm looking at there is a cell, and in the middle of that cell is the DNA in the nucleus that's in there. It kind of looks like a fried egg. And then as we move on, I want to move down and look at how that DNA would look inside there. And so this next drawing of the cell here, which we can see, is a larger cell membrane. I've enlarged this nuclear membrane inside so you can see that that's there. And then I've drawn a chromosome of DNA in there. And so, to be fair, when a chromosome begins, it might actually look like bloop, like a lot smaller, like this just little squiggly guy there, right? So I've just got this guy. And when the DNA replicates from a single chromosome, then it actually makes a second long strand, exactly the same, and then they're joined together in this thing, connected, this protein mass, we might call it, which is called a centromere. And the centromere is a pretty important term because it's this glue that holds these two things together. And so the centromere, and you can see it down here in the next images below, that here I've got one chromosome, right? But now you look over here and you've got the same thing. You got the duplicated DNA. It's replicated, but held together in the middle by that centromere. This is this key function because you can imagine now that if you wanted to replicate all the DNA in an organism, each single chromosome replicates itself, and it's held together in the center by this centromere. And then if you just tear apart the two sister chromatids, we call them, and you tear them apart at the centromere, then you have two exact copies. So all you need to do is do this for every chromosome and tear them all apart at the centromere. And that is the process by which cells undergo the process of mitosis, which is to replicate. So let's take a look at that in the DNA. So here's an image of my DNA here, and I've got basically this big thing here is supposed to be a hunk of DNA. It goes on for millions of bases in each direction. But the way that DNA might actually replicate is that it would be opened up like you have in this drawing. I made it open up right here, and then it has a split, right? So then you get a space in the middle, and it might get opened up by an enzyme, so we can write that in there, opened by an enzyme. And there is a specific enzyme that does that. And then another enzyme, and this is where you see all this red drawing on here. This red drawing represents like the big donut. I know this stuff here is a big donut. It's kind of hard to show you what that would look like. I think they probably have some images online. If that's your DNA, the enzyme I'm interested in called polymerase actually looks like this. It's a big donut that wraps around the DNA, and then it can slide down the DNA. And as it slides down the DNA in this open position, it fills in all of these extra bases here that are complementary to the original bases that are there on the original piece of DNA. So what results from this is the DNA strand gets ripped apart. If you imagine breaking a ladder along the rungs in the middle, so that basically gets cut down the middle, and the result, right, is that you have the original DNA here and the original DNA here, and they're basically two halves of the broken ladder now. And then what you've done is you put in new DNA on either side. And so now you have two exact copies. And this is the sort of fundamental thing of DNA. This is what makes it amazing. It can be copied millions and millions of bases without mistakes. Some mistakes are made, but very, very few. The enzyme that does this is called polymerase, so you'll hear me referring to that. Cool, so that is DNA replication. So when we talk about mitosis, what you would do then is line up these replicated chromosomes. So this would be like replicated DNA. So I can write that on there, right? That's replicated DNA. And if you line them all up on their centromeres and just tear them apart at their centromeres and put them into different cells. The way the tearing apart part is done is these little green lines that are on here. Guess I can keep them green. These little things are called spindle fibers. And those spindle fibers actually form like small cables, and they attach to the pieces of DNA and they pull. And when they pull, they actually pull them apart. And there's some other things that help disintegrate that centromere, but that's essentially you're ripping apart at the centromere. Let's take a look at what's next on here. We're not looking at this right now, so I'm going to erase that stuff. This is the important part that I just finished right there, equal amounts of DNA. Now, most organisms are diploid, which means they have two of each chromosome. And that means that what they would look like is more, if I could draw that for you right here, which I'll do, I'll make a diploid-looking organism. It means that one chromosome would look like that, and they have two of the same. They might have another chromosome that's short, another one that's short. So these are two different kinds of kind. This is like kind 1. This is kind number 2. And they have two copies, but they are not exactly the same. And we call them homologous, but they're not identical. And what I mean by that is that each of them may carry a gene, like a gene, and I use this in the demonstration below. For hair color is the gene, right, which is the gene, but then I have the same gene is in the same place on the other one. This one also has the hair color gene, right? You see that it's on there. The difference is this one might code for brown, and this one might code for blonde. And that sort of difference is what we call alleles. So the alleles are alternate forms of the gene. So it basically, while the gene itself is the hair color gene, the actual color can vary from one homologous chromosome to the other. So when that DNA replicates, what does it look like? It looks like this, right? And this one replicates, and it looks like this. And so now you can see that the replicated DNA looks a little bit different in this diploid organism, and I've got them lined up so they're with their homologs. But let's go on down here to the next image. This is exactly what I was just talking about. And in this is more common for my YouTube videos. I like to represent the two homologous pairs, and I'll circle those for you here. those two homologous pairs, right? Like these, this is a pair, this is a pair, this is a pair. And I put them next to each other and I distinguish them by making one of them stripy, like this guy, and I make the other one clear. And what that indicates is that one of these homologous chromosomes came from the mother, one came from the father. They do have different origins, and they're going to have different alleles, but they're going to have the same genes, right? So eye color is going to be the same place on, a chromosome number 1 on both types, right? So I distinguish different types or kinds, right, by the length, and that's writing up here, and I do different origins, or which one is which of the two homologs, as stripy or not. So humans have 23 homologous pairs in the human, which is 46 chromosomes total. As I will mention later in the lecture, the X and Y chromosome are the 23rd pair, and they're not precisely homologous for the entire chromosome, so they are a little bit different. And we'll talk about that later in class. Is the 23rd set. So you know now that a gene is a region of DNA that codes for a trait, and allele is an alternate form of a gene. So one of the things I like to look at in class is to show that some traits are sorted out by this sort of simple dominance interaction, right? Which means that here's the trait that I have today. I made these little drawings for you guys. This is your thumb, and it's called Hitchhiker's thumb. And so if your thumb is curved, when you put it up, curves backwards, they call it the Hitchhiker thumb. And then if other people have a thumb that when you put it up, it just stays up straight. Neither one of these are, they're both perfectly healthy thumb shapes, but they do differ because of it's either the length of the tendon or the way that the joint is formed in there that makes these different, but it appears to be controlled by a single gene, right? So that means that the thumb shape gene is the gene, and then, so we can call it thumb shape, and then you can have either curved or you can have straight thumb alleles. So now, when we have cell replication, and let's take a look at this down here, I'm going to go through mitosis, and we can look and see how this works. Remember, when cell replication occurs from that first parent cell of the zygote, you get right here, I could label this the zygote, if you want, and then the descendant cell is two identical cells. Now, the only difference between those two cells is any mutations that might occur in replication, but those are relatively rare. We're talking one in a million, maybe even 1 in a billion bases. Is there actually a mistake? So we go through the mitosis. I'm going to stop here. Ka-ching. Break. And I'm going to end the video, and then I'll start the next one.